Hi. RNA splicing has, through in the form or through alternative splicing, we have seen how one gene can give rise to diverse proteins or more than one type of protein, and that increases the diversity of the proteins that can perform diverse functions and thereby resulting in complexity. There is also one another way of how one gene can give rise to more than one type of proteins, and that is called as RNA editing. When we say RNA editing, it's all about changes in the sequences of the bases. Upon production of the mRNA, say mature mRNA, because there have been changes in the base sequence, we will have consequently the proteins as well, the changes. And that's what we will try to see now. This is called as RNA editing. We are making informational editing of the mRNA on the mRNA, resulting in production of different types of proteins. There are two ways. One of it is kind of substitutions in a way. That is, you're changing the base sequence by altering some groups. So you're not actually changing the number of bases. You're only changing the composition, the nitrogenous base. It's not a prevalent one, or it's not everywhere that you would find, but whatever are possible are something that can be done. So by base modification is one. The number of bases will remain the same, but we'll see that the sequence of the base has changed, at least in some locations. Like, it is kind of analogous to point mutations, like substitutions, like transitions and transversions that we may have studied in DNA repair, damage and repair. There is also another way where some bases can be added into the mRNA. There you will see that there is increase in the number of bases itself. Okay, the first one by modification of the bases, and here we will see an example, and here is a pre-mRNA, and we have a codon 2153, which is a huge protein actually, and in one of the exons, in I think it's exon 26 or so, and codon 2153 is present. And in this gene, we would see that this is a gene for a polyprotein, and it is present in or expressed in two different tissues, mostly in liver and intestine, and it is important for transport of lipids across through the bloodstream from various organs. Right now, we'll focus on what happens in light of RNA editing. So the mRNA, and this mRNA is spliced, and so we would have had a mature mRNA. And in two different tissues, say we'll compare that what happens to it in liver and what happens to it in intestine. In the liver, there is no RNA editing. There are no bases changed. What you will see is that the translation of the mRNA would occur, and CEE codes for glutamine, and we will have a protein of about 4,500 amino acids. In the liver, in the intestine, what happens is that the the cytosine that is present here, it must be a, say, CAA, and this cytosine could get deaminated, resulting in uracil. And because CAA is a sense codon coding for glutamine, whereas UAA is a stop codon, and therefore upon translation, this mRNA will give rise to a shorter protein of about 2,100 amino acids. If we compare to what happens with and without editing, it would have been about 4,500 amino acid residues, whereas the one after editing will only produce around 2,000 amino acid residues. So these are carried out. Here, how did cytosine become uridine? And there is an enzyme called as cytidine deaminase. So the amino group that is present here would have been removed. That would have been carbon number 1, 2, 3, and 4th. There is an amino group on the 4th carbon, and that amino group is replaced with a keto group like this. Now, deamination of cytosine will result in the formation of uracil. As we've seen also in DNA damage, where spontaneous deamination of cytosine could occur, resulting in the formation of uracil. There we have uracil glycosylase to eliminate all uracils from the DNA, because uracil is not a part of normal basis, nitrogenous basis in the DNA. Whereas in RNA, uracil is a normal base to be. And that is how a sense codon that is CAA, which was coding for glutamine, could have resulted in formation of UAA, a stop codon. And this is not, of course, a mutation. It is not, because it is happening in the in the RNA. And if it were any base sequence changes that happen at the level of DNA, we would have called that as mutation. But at RNA level, we don't call it as mutation. So there are also other enzymes which can specifically identify a particular base and only act on that base. Again, cytidine deaminase would not go and randomly deaminate all the cytosines. They are highly regulated and coordinated structures or events. There is also another enzyme called as adenine deaminase acting on RNA, or in short you call it as ADAR. And so it's similar to that of cytidine deaminase. The ADAR also deaminates the sixth group, the amino group present on the 6th carbon, thereby resulting in formation of inosine. Inosine is a kind of precursor for all the purines. Here, inosine is not a normal base in the RNA as such. The one thing is adenine would have been base paired with thymine, whereas inosine could base pair with multiple others, including a cytosine. So probably this would have a function in the way the RNA folds upon itself, because now the inosine could base pair with cytosine. It could result in a different three-dimensional structure, and thereby may affect the functions of that RNA. And there is another type of how the RNA editing could be done. What we have seen until now is changing the base by modification. We're changing one base to become another, like cytidine to uridine, adenine to inosine, etc. We also have other ones, that is by addition of bases. This is much more complex and quite interesting as well. So here we have something called as guide RNA. Guide RNA is, you can say, as the one that dictates where and what should happen in a particular mRNA. So it is highly based on sequence specificity or sequence complementarity. So guide RNA has 3 components. One of it is called the anchor. The other one is the editing region, and then we have a poly-U stretch, multiple uridines that are present. So the anchor determines where addition of a particular base should happen. So that is the function of it, where it should happen. Editing region, of course, or you can say to which mRNA there should be. So based on the sequence homology between these two, things will occur. And in the editing region, where exactly and what should happen is determined by the editing region. And then we have poly-U stretch for multiple other functions. So what happens here is the guide RNA base pairs with the target mRNA, and of course by base pairing we are saying that we are inducing certain degree of specificity, that because of sequence homology or sequence complementarity, it's able to bind to the target RNA. And if we notice it carefully, say in this case, we have complementarity absolutely as it is, say G, AU, and then we have GC, AU, etc. The other two adenines were added already. We are just showing what is there. Addition of two other adenines that are present. So the guide RNA is almost the lower, this is the guide RNA. It's almost in this particular region, almost complementary, except for these two adenines. The two adenines are bulged out like this. So in that location, we have an endonuclease that will act on the mRNA, and it will break exactly in this place where the two adenines are bulging out. And now we have a 3' OH and 5' phosphate, and there is a space because now the guide RNA could stretch out a little bit, and there is an RNA there is an enzyme called as TUTase, TUT, terminal uridyl transferase. And what it does is, to the uridines that are present, so to the 3' OH here, it would add uridines in this location. So it will add two adenines. There is no more space than that to add. And the two adenines are added by TUTase, again, terminal 3' terminal uridyl transferase. All the function is said by the name itself. It is taking the 3' OH, 3' terminus, uridyl, which means that it is adding nucleotides of uridine bases. Okay, if it is just addition of a nitrogen base alone, you can probably call this uridine transferase. When you say uridyl transferase, then it would mean that it's a nucleotide. So it would add the two uridines in this location. I mean, it's transferring. So that is what it does, and there is no more space. This is from here to here, it is continuous, and we would still need the nick that is present in this location to be filled up, and that is done by an enzyme called as RNA ligase. And now the original mRNA had a sequence GA, GU, AU, ACCU. And these now, upon editing, we have two different uridines. I hope you can recollect the significance of change in the number of bases in an open reading frame. It would change the reading frame itself. It is something similar to what would happen in a frame shift mutation. So until here, the protein could probably be coded with produce amino acids residues or add amino acid residues accordingly. But here, upon addition of these uridines, the whole reading frame has changed, and the protein that is going to be produced will, of course, be different, at least from the site of the addition of these bases. What is frame shift mutations? When you have deletions or additions which are not multiples of 3, the whole reading frame is going to change. Almost the same thing is happening here, except that it is happening in DNA. It's happening here. RNA editing is being done at the level of RNA, and it's not the genome, so it's not a mutation. It is just producing a different type of protein because of RNA editing. It's wonderful to know that there are many more things, and as a part of science, we will keep on getting to know many more wondrous things that happen in the biology of all living organisms. See you later. Thank you.